loud. Okay, it's all yours, Tae Wu.
Oh, thank you for a nice introduction, Irina. First, I'd like to say thank you for official invitation of very nice Zoom talk in biocars. So my name is Tae Wu Kim from the Mokpo National University, South Korea. Today, I'm going to talk about the Deciphering structural dynamics in by using the time-reserved access solution scaling. I will give the very small introduction for that. If we closely look at the water in the Michigan Lake, then we can see the very clear structure, molecular structure of water like this, and its H-bonding network. If we have some time, and we will closely look at the tracking, there's some real time, some change of this water, then we can see this kind of the dynamics of water structure and its related some environment change. So this way, the protein has some molecular structure and its relative some motion is very important for our chemistry. So this kind of theme is also working in the biology. So the structure and the protein structure and its related motion in biomolecule is very important. So we should well know about that. So this is the one of the example. That's the amyloid beta peptide. This very well-known peptide in the biology. And under the certain situation, this peptide can be dissociated from the original structure like this. So this is the MD simulation, for example. So this kind of phenomena is called as the unfolding motion of peptide. So such a protein can be chemically equilibrate between the unfolded and folded state between them. So some case, this unfolded structure and conformation can be aggregate one another. So it can make such a unwanted aggregate form that's called as an amyloid flag. So this flag can block the signaling process in the neurons. So it finally results in some disease from the protein disorder. So this kind of phenomena, we don't want that. So that's why we should know about the protein structural dynamics and its dynamic behavior like this. So I'd like to say that protein structure and its dynamic motion, it can leverage the biological function in our living organism. That's why we should know about that. We should understand about that. So today my talk is constant of the two subjects. So first one is the protein folding dynamic study in cytochrome C, and second one is another signaling protein example. So first subject is the study about the protein folding in cytochrome C. Let me put it this way. The unfolded conformation can be chemically equilibrate with the disfolded conformation like this. So this upward conformation going forward, also this protein conformation also can be going backward. So these two conformation is well equilibrate under the certain situation. Some case, such an incorrect protein folding can be happen. So in terms of the protein structure function relationship, this kind of incorrect folding, we don't want to do that. So this incorrect folding can make the unwanted disease in living organisms. So in our study, we want to directly track the real-time change of a protein during the protein folding mechanism. To do that, we use the very special probe, that's the X-ray solution scaling. This is also known as the SOX wax measurement. So we all know, we are all familiar with this kind of measurement. So if we collect this kind of very high quality scaling pattern like this, from this pattern, we could get the direct structure information. So in this case, we can get the protein structure information from this scaling curve. So this X-ray scaling measurement can be combined to the time-reserved experimental manner. So we called this measurement time-reserved exercise scaling. So I will call it simply TRXS. So time-reserved scaling. It is based on the pump and probe geometry. So in this experiment, the optical pumpers, it can initiate the photoreaction of the target molecule. In our case, that's the protein. So another XF pulse, it will be used to record some scattering pattern as a function of the certain pump probe time delay between the pump laser pulse and probe laser pulse. From this way, we can get the time reserved the different scattering curve like this. So if we interpret this different scattering pattern, then we can finally get the information about the protein structure and its motion. So my target system is the very well-known protein. That's the post-hurt cytochrome C. This cytochrome C can be acting as an electron mediator in our living organism. So we are all familiar with this protein. This protein has some advantage for our study. First, if we use a very special reagent, that's the NADH, then after that, if we use the UV light, so the irradiation UV light into this NADH, it causes the photoreduction into the oxidized unfolded cytochrome C. So from this way, this unfolded state or unfolded conformation can undergo some spontaneous folding toward the folded state. So from this way, we can initiate some photo-induced folding in cytochrome C. So from this way, we want to track the real-time tracking of this protein folding dynamics by using the time-reserved accessory scattering. Before starting the actual time-reserved experiment, we first characterize the steady state structure for oxidized unfolded and reduced folded cytochrome C. To do that, we adjust the chemical environment for the cytochrome solution. So such as we adjust some inertial condition, or we also control some redox state in here. So from this way, we chemically made some starting oxidized state and reduced final state. This is some actual data for static X-ray scattering measurement. So left bottom panel shows this data shows the reduced folded form data. So to get the structural information from this pattern, I use the NMR, solution-based NMR structure. This is very well-known structure. So from this way, I could get the theoretical X-ray pattern from this structure. So as you can see in this graph, the red line shows the theoretically calculated scattering pattern. It shows the great agreement between the experimental and theoretical. So from this way, we can say that this kind of NMR structure will be the optimal structure in our reduced for this state. By using the same analysis, we tried to reproduce this oxidized unfolded state structure, but we could not get that. So I simply showed one of the trajectory for unfolded conformation of cytochrome C. Based on the various type of unfolded conformation of cytochrome C, we calculate the various type of theoretical scattering pattern, but any single protein structure cannot explain this experiment pattern. It means that this oxidized unfolded state might adapt to some very not single protein conformation, but it will take the various type of conformation. So to do the structural analysis for this data, I changed our analysis scheme to the new one. That's the ensemble-based analysis for that. Let's think about some protein conformation in the unfolded state. It will be like this, such as this blue extended structure or lead extended structure with some magenta structure. All of those shows a very extended conformation like this. Also, all of that will not be same. So it means in our unfolded conformation, the conformation homogeneity will not be working in this situation. So to analyze this data, we should adapt the new philosophy. So that's the ensemble-based structure. This ensemble analysis is based on by considering the conformation heterogeneity for the very extended unfolded conformation. From this way, to make some theoretical scattering curve, we mix all of the unfolded state scattering curve with those latent populations. So by considering both of them, we can get some theoretical scattering curve based on this conformation heterogeneity. From this way, we use this actual layout for the analysis of our observed the collected scattering data. First, high temperature MD simulation. From that, we generate the various type of unfolded structure candidate like this. So this structure shows the very example structure, but this structure shows a very extended protein conformation, and also this MD simulation generate the partially unfolded conformation. So we collect all the trajectory from there. So all of those structure could be used for the theoretical calculation to generate to make the theoretical scattering curve like this. So based on those theoretical scattering pattern, we could do the additional analysis like this. So I will show the actual result from that. So this light panel shows the ensemble-based analysis result. As you can see in this picture, so the reduced state shows very homogeneous populated state as a function of the rate of gyration. So this state shows the very well-organized folded structure. It shows that. But in the unfolded, oxide unfolded state, it shows a very heterogeneous populated ensemble information as a function of the rate of gyration. So this below structure shows the representative conformation about this unfolded state. As you can see in this picture, so there are many different extended conformation like the partially unfolded structure and quite extended structure and very strange structure. All of those are in the unfolded state. Based on our observation in the static X-ray scattering measurement, we performed the time-measured X-ray scattering study. As you can see in this actual data, there are very clear oscillation pattern in the smoothing region. Also, this pattern, this picture can be formed around the 10 milliseconds. So we can say that maybe some global conformation change is happen during the protein folding dynamics. We can expect that. However, this entire spectrum a little bit hard to understand or digest. So to extract the species associated spectrum for each species, we did the global kinds analysis to get some species specific character from this data. So as you can see in the light panel, this red line shows a second species associated scattering spectra. You can see a very clear picture like this. So I compared this experiment data with the static difference. This static difference is from between the fold and unfold state. So as you can see, this time-measured data shows a very good, quite good agreement with the static difference. it means the second species can be assigned to the folded conformation. So it means around the very late millisecond time delay, the folded form can be generated in our dynamic process. We closely look at the population dynamics of the cytosine folding state. So this red curve shows the folded state. So the rise of this folded state shows a very strange feature. What I mean, this green one shows the normal exponential kinetics. If we take a look at the PChem, physical chemistry textbook, we can map the very simple unimolecular reaction like this. If there are some unimolecular reaction from transition from the A state to B state, usually this kinetics, it can follow the first order kinetics with the exponential decay. But in our observation, this folding decay profile shows a huge deviation from the normal exponential decay. So this red line follows the very strange stretched exponential kinetics like this. So what's the meaning of it? If you ask me what's the meaning of this stretched exponential kinetics, in this kinetics, there are many different folding pathways. All of those are superimposed in the kinetic wise. So this very heterogeneous population transition can be expressed as a very special type of exponential. Actually, this result can be connected to our result from the structural analysis for the steady state x-scale measurement. So initially, the heterogeneous populated unfolded conformation, they can adapt the different folding kinetics. So all of those can be mixed in our observation. Then these folding dynamics can be expressed as the very strange exponential kinetics, not the normal kinetic dynamics. So I'd like to highlight that the cytometry folding include many different folding pathways with different folding times. So that's why we found the very strange kinetics in the folding dynamics. So this heterogeneous populated unfolding conformation, it will follow the different folding pathway. Actually, this scheme can imply the funnel-like folding model in the biochemistry textbook. So in the funnel-like folding model, in unfolded state, it considers the heterogeneity of protein conformation. So many different unfolded conformation, it can take the different folding pathway. So maybe our observation will be one of the experimental epitome for the funnel-like folding model. So if I recap my first section, this heterogeneous populated unfolded conformation can take the different folding pathway with different folding time towards some singly homogeneous populated some native state. This kind of funnel-like folding model, it resembles this whirlpool-like picture in this figure. So this is the end of the first section. I will move to the next part. That's the study about the light-induced protein dynamics in bacterial phytochrome. Bacterial phytochrome is one of the well-known photoreceptor in living organism. So in our living organisms, such as the plant, bacteria, and fungi, etc., in there, this phytochrome, it can regulate the wide range of biological signaling process. So this biological signal pathway mechanism, it includes some isomerization of the bilirubin chromophore like this. So some study reported that in this process, it has the two different transient species. That's the PFR state, another one is PR state. They are major component during the signaling mechanism in phytochrome. So my target system is among the various type of the phytochrome, I focus on the bacterial phytochrome, not plant, bacterial phytochrome. You may know the Professor Xiaojing Yang in UIC and Keith Mokpo in University of Chicago. They are first studied this bacterial phytochrome for the docu-incubated PFR state. So they are found that this docu-incubated PFR state has the very parallel aligned helical backbone like this. So this is a very well organized helical backbone in this system. In that study, they reported that this bacterial phytochrome has the parent state, that's the PFR state. And photoexcitation from here, it can generate the final light-induced PR state, that's the final product state. However, such a far-induced photo chemistry in this phytochrome, it is still unknown. So my study is to focus on the real-time tracking to study this phytochrome dynamics and its relative structural motion by using the time-zero exertion scaling. This is the actual experimental layout for our study. Almost scheme is similar to the setup used in the protein dynamics, but one difference between the previous study and current study, that's we use the additional light with to the dump light, acting as a dump light. This bacterial phytochrome has the two different light-induced state, that's the PFR and PR. So we want to make the very constant chemical equilibrium between the PFR and PR. That's why we use the additional light to keep some constant population between the PFR and PR. That's why we use that. And experiment detail I will summarize here. So all of the experimental implemented in the biocourse group. This is the real-time experiment data. So as you can see in the light panel, you can see a very clear some oscillation picture and each change in the SOX region. So from this data, we did some kinetic analysis. It also very similar to the analysis scheme I used in the protein for the dynamic study. So from this kind of study, I could find some kinetics in working in this phytochrome protein. The phytochrome has the three different species. That's the I1, I2, and P state. So this I1 state can be converted to the P state with by a I2 state, and this working mechanism has the two different time constants. The first one is the 90 microsecond, and second one is the 21 millisecond. Also, these spectra show the species associated spectra. So you can, as you can see in this slide, you can see the very clear spectral chain, spectral difference scattering curve in SAGE2 and SAGE3. Based on this spectra, I did the additional some structural analysis based on our collective scattering curve. Before showing the result from the structural analysis, I can say that I used the three different candidate conformation for bacterial phytochrome because there are some previous X-ray crystallography. They suggest the three different conformation may be working in phytochrome. That's why I use a three different conformation like this. So a little bit hard to pronunciation. So I simply describe the three conformation like this. Two helical backbone in photosensory module, they are almost parallel aligned like this. So I just call this conformation like the II plane. And middle one shows a very some round type helical backbone. So I just say that this is the O-framed geometry. And final one shows a very stretched conformation like this. So I will say that exactly this is reversed Y-frame, but I will simply call it the Y-frame geometry. So from this conformation, I could make the various type of molecular dynamics generated conformation. From that, I can do the pairwise fitting for times x-scaling curve. From this way, I could find the optimal structure for bacterial phytochrome. As you can see in this picture, the I2, the second species has the very parallel aligned helical backbone. That's very similar to the structure of PFR state. That's our starting point. On the other hand, the P state shows a huge conformation change it undergoes. So as you can see in this picture, the helical backbone is very strength. So it shows very O-frame structure. So we can say that P state has a very O-frame structure. So let's imagine the photocycle of this bacterial phytochrome. Initially, this PFR state with a very highly parallel aligned helical backbone, it can be bent during the formation of P state. So from this way, we could find the optimal structure in phytochrome. Based on our observation, we could make some photocycle for the bacterial phytochrome. However, I'd like to say that we want to add the origin of the transient species in terms of the chromosome structure. So we use the previous spectroscopy result like this. So if you simply compare between them, the time constant observed in the TRXX, they are very similar to the result from the optical spectroscopy. So we could assign what's the exact origin for each species. So from this way, I2 state can be correspond to the meta F detected in the spectroscopy result. Also, the final P state can be correspond to the PR state. That's our final photoproduct. From this way, we could make the final structural dynamics and its photocycle like this. This is the end of what I prepared today. So by using the time-reserved accessory scattering, from this way, we can investigate the photoinduced structural dynamic in the various type of protein. So, the X-ray scattering probe, it will be very powerful tool to reveal the protein structure information for our phytochrome or cytochrome C or something like that. So from this way, we could study the various type of protein. I'd like to especially say thank you for my co-workers. First, I'd like to really appreciate my PhD advisor because the Professor he guided me for this protein for study and many of the protein studies. So really appreciate his advice. Also, I'd like to especially thank you for my long-standing collaborator, who is the BioCast member, Bukija, Irina, and Robert Henning. So I really appreciate their extensive support during my graduate school student in KITE. So thank you for your attention.